Hello, and welcome back to GlycanHub, the podcast in which we explore health, disease, and longevity through the lens of glycobiology. My name is Rina, and I am your host. Omics is a rapidly evolving, multidisciplinary field which allows us to gain a comprehensive understanding of the underlying structure, function, and dynamics of a biological system. You might be familiar with some of the omics layers, such as genomics, proteomics, metabolomics, and so on. However, a multi-omics approach takes it a step further. By integrating the data from separate omics layers, we are now able to generate a multidimensional view of human biology that unveils the complex interplay of networks within our system. My guest today will introduce us to the current state of multi-omics research and its potential to revolutionize the way we understand, diagnose, and treat diseases. Listen in to find out why glycans are considered intermediate phenotypes and how this makes them promising biomarkers, as well as why glycans are often overlooked in multi-omics research. In today's episode, I am joined by two scientists from Weill Cornell Medicine in Qatar, Dr. Karsten Suhre, the Professor of Physiology and Biophysics and the Director of Bioinformatics Core, and Dr. Ania Halama, who is an Assistant Professor in Research of Physiology and Biophysics. Karsten is a pioneer in multi-omics GWAS study. He shaped the field with his contribution to metabolomics and proteomics GWAS by highlighting their role in human health. His research focuses on complex diseases where integrative omics analysis could provide further insight into disease mechanisms and treatment strategies. Ania focuses predominantly on dysregulated cancer metabolism and its role in drug resistance. She's working towards strategies enabling the implementation of metabolomics and other omics technologies into clinical pipelines to support future diagnoses and optimize treatment modalities. A warm welcome to Ania Halama and Karsten Suhre. Hello and welcome to our podcast.
Yeah, hello. Hi.
Hi, hello.
So, unlike the majority of our other guests, you two are not glycobiologists. So, could you tell us what it is that you do and how do you know each other?
Maybe I should start on that.
Yeah.
Because actually, Ania was, I was her co-supervisor in her PhD, like I think you said 15 years back.
Yes.
It's scary.
We don't want to count the timing because it starts to be scary. You know, it's fine when it's for 1 or 2 or 3 years, but then 15 years, you are getting like, well.
Yeah. No, we started together at the Helmholtz Center in Munich. And that's also where we both started, I think, on metabolomics. I was more on the bioinformatics side and Ania is more on the wet lab side. And that's how things, I think, then I moved to Qatar and Ania did her PhD and I asked her to come. And maybe you can take it from there.
Yeah, I think so. The story starts at Helmholtz, as Karsten is saying. So it was amazing experience. You know, we were always interacting together, and in fact, Karsten kind of determined the next steps in my career because I decided to follow him here and to make the work in his lab. So the initial work was focused on metabolomics because this was the specialty I was trained during my PhD, and this was also the specialty of Karsten. So he was having this virtual metabolomics lab, and I was trying to add the wet lab component where we could, I don't know, look at certain things, checking for the mechanisms and things like this. So this is our story. This is how we started to work together, and we are working until now, and it's like 15 years all together when we counted it, which was kind of interesting, you know, if you think about the years.
Oh, that sounds like you get along well.
Yes, because, you know, usually it's something like, I mean, you're having many collaborators, but I think it's very cool.
Yeah, but we try to make it complementary because I have two left hands for whatever is coming to a pipette. And Ania is very good at working in the lab and with cell culture and samples and everything. So I think it's the complementarity that makes it.
Excellent. And in today's conversation, we are going to focus on metabolomics, but specifically also more on looking at a bigger picture and looking at a multi-omic approach to research. But starting with metabolism, we have in our everyday life now started to use the word metabolism pretty frequently. So do you think people tend to misunderstand what is meant by that word and how it works?
I think people are using frequently metabolism. Start to do exercises, you will gonna turn up your metabolisms, change the diet, you will gonna reduce weight because you are changing something in your metabolism. And I think in that particular context, they are using the word metabolism very correctly, right? And they understand pretty well what metabolism is, because everybody was having on some point in their life biochemistry classes when we were talking about, starvation or making exercises and things like this. But we need to think about two different things. On one hand, we are having metabolism, right? When we are looking at the function of the body. On the other, we are having metabolomics, right? And metabolomics is in fact the study which is enabling us to provide further insight into this metabolism, right? So it's a very powerful tool. And I think people are not really using metabolomics as a word because not many people, like in general population, heard about it. But metabolism as such, yes, they are using frequently. In my view, they are using it correctly, right? So that's how I see it. I don't know how you see it, Karsten.
Yeah, no, absolutely.
That's a relief that we actually are understanding in everyday life what metabolism is. But could you define more closely what metabolomics means?
If you think about metabolomics, it's a pool of metabolites, right? So ideally, you will gonna be looking for all the small molecules in the sample, right? So not only on glucose or lipids level, which you can do with the simple biochemistry, but looking at the all in the same time. So this is how I see metabolomics. So studying the small molecule composition in the given sample, and whatever the sample is, it can be human sample, it can be dates, I mean the story on dates, or it can be everything, whatever you can think of. So
it's just like genomics, proteomics, metabolomics. You just do metabolism on a whole, and of course it gets more and more complicated, right? In genomics you have just 4 letters, in proteomics you have 20 letters, amino acids. And in metabolomics, the problem is like every molecule basically, not every molecule, you have acids and bases and all that kind of things, but it gets even more complicated, and that makes the measurement much more challenging. So there's not one tool to measure all metabolites. There's more or less one tool to measure all proteins in proteomics, and there's certainly one tool to do all genomics. So the more you go towards metabolomics, the more complicated things become in terms of measurement.
Could you perhaps give a small overview of how the different omic layers interact or relate to each other?
We are having obviously all the omics, which Karsten was just mentioning, right? So in fact, they are linked together, right? Because you are having from genomics, you are going to transcriptomics because of the transcription, right? The transcripts are later on translated to proteins. Proteins are enzymes, transporters, name it. And then metabolites are products of the substrate for function of those proteins, right? So this is the way how all of those are interlinked, right? Then is another factor, which is not all enzymes are gonna be working in the same way, and they are sometimes changed. For example, with glycans, right? So we are having glycosylation, and then this glycosylation might change the function of the enzyme. Also, if you think from the perspective of genomics, right, you can turn on or turn off the genes using methylation, right? So it will be another way adding the complexity to all the omics layers which are together. Like, so I would say that everything connects with everything else, which makes it fascinating in one way. In the other, if you think about the complex diseases, it's pretty difficult to interpret later on and work with those data, right? Yeah. So this is how I would
and some of our early collaborators from the company Biocritis, they had at some point this image thing to say, like, in the end, the more phenotype you get, the more it's like imaging, right? In the end, every single omics phenotype is a pixel in a picture, and the more you get, the sharper the picture becomes.
You have mentioned Glycomics as being one of the omic layers. Why do you think it is often overlooked when in studies that are taking a multi-omic approach?
It's not that popular. I mean, think about the collaborators, right? And think about possibilities. Right now, metabolomics, it's very easy to think about, right? It's metabolites. Everybody knows about glucose. Everybody knows about lipids. Amino acids, right? This is something pretty natural to add to your studies, right? But not many people are aware of glycans, right? And also, if you think about technology, the aspects and the collaborative work, I mean, there are not many labs which are specifically focused on glycomics. You need to have extra knowledge to do it, right? So metabolomics right now, people can do on their own, right? There are some kits which you can implement, you can do metabolic work, you can also try to do something very easy for yourself. But with Glycomics, I feel it's more complicated, and I think that this is one of the reasons of this complexity to the story.
Right, and also in metabolism, you have like 150 years of biochemistry already going on.
Yeah.
Even if people know about glycans, I mean, Gordon Lauch is, I think, very outspoken about the Human Glycome Project, and people really underestimating the role of glycans. I totally agree on that, but the limitation is really how do you measure this in a straightforward way. We got some data actually from Gordon, but I mean, it's still very limited, right? You just have glycosylation, for instance, only of one protein, of IgG, for instance, or you can get all the N glycans, but that would be just 30 N glycans on any protein. So it's very hard to measure everything in, especially if you go for larger studies, population studies where you have clinical outcome. So you may look at this in much detail if you do a study in a cell culture or if you look at a specific process, but to do it at the population level where you would work like what we do with proteins and metabolites and genes, I think the biggest limitation at the moment is the measurement, to really get this data, to really show what the impact is. And I think Gordon has done a great job already showing examples, but I think there's much more to come.
Yeah, that is a very common answer when I ask a similar question, why glycans aren't more researched. It is the technological limitations.
Yeah.
Now, segueing to the molecular human, could you tell me about the Qatar Metabolomics study on diabetes? What were you initially aiming to do with this study, and how has it grown into the molecular human?
I came to Weill Cornell Qatar like almost 12 years back. And at that time, I came from Munich, so we have been doing metabolomics and all that kind of things there. So the idea would be we would like to do it here again. But then the challenge was, where are the samples? So there was no Qatar Biobank at the time, and no Cora study and no Framingham study for Qatar. So we started what we call the Qatar Metabolomics study on diabetes, QM diet. Initially with the aim to send it to Metabolon, because that's the big study we did at the time. And we said we collect urine, saliva, plasma, which was a little bit, it was a bit inspired at the time. Qatar Biobank was just building up, and my colleagues from Munich were coming here to consult with them, and also people from Imperial College were here to basically build this up. And we said maybe do a little kind of a pilot for a biobank study. And so we said we do a diabetes study, because that was one of the priorities in Qatar at the time, because there's a high diabetes rate, as you may know. So we said we go and collect 50 samples cases, 50 samples controls, because we just had done that in Cora before in Munich. And we were just over lucky that in the end we didn't turn out with 50-50, but with basically almost 400 samples. And we also, it was really serendipitous in a way, because we wanted to do that with the diabetes department, but there was no space. They were so busy. So in the end, we turned out at the dermatology department, because there are many diabetic people with skin disorders, but you also have a lot of people who have minor things that you could actually use as controls. And we really collected almost 400 people, totally mixed. So there was really no batch between like cases and controls collected different places. And I think that's very important, and we were lucky with that. And we created, I don't know how many tens of aliquots per individual. Initially, we just wanted to do metabolomics, and then really over the last 10 years, every time a new technique came up, we said, oh, let's just try it on this pilot. So we have a great genomics core here. They ran genotyping arrays, methylation arrays. We did whole RNA sequencing on them. And then at some point, I was in contact with Gordon Lauch, who basically was already doing the measurements in Cora. And we just say, oh, why not send them to Gordon as well? And then maybe I'll let Ania continue with that, because we have a lot of more data in there. And Ania is just presently in the trying to integrate that into one the molecular human. Yeah. So maybe go on with that one.
Yeah, story is like this, and also it will be important to mention that that time the diabetes information on diabetes and diabetic project was not so. There was not so many studies at the time when you started to work on diabetes, metabolomics and stuff, because it was 2013 when you get the data, no?
Yeah.
So there were also not so many information about diabetes in terms of metabolomics, and that time was also the new Metabolism platform where the study was run. So all of this, was touching upon the technical development as well, right? But I think once the O-link came to the market and we tested as a new technology also O-link from just curiosity, we started to look at the data in more like the way where we are trying to compare like two different technologies, where we are looking at different data sets. Of course, the methylation with diabetes and stuff, all of those publications were coming down the line, right? So there was many like PWAS papers from Shaza, for instance, and all others. But on one point, we look at this one and said, well, I mean, it's kind of very first study which is looking at so many omics in the same time, because besides
how many do we have now?
We have 18 platforms, right?
Because I think what's maybe also important to know is, I mean, here Weill Cornell in Qatar is very new compared to the hundreds of years of history of Cornell. Is very new. I mean, I think the research started in 2010, and we have something which is very important, our different cores, genomics cores, proteomics cores, bioinformatics, metabolomics cores, and we use this study basically as a trial bed for all the different tools. We have a SomaLogic platform at some point. We brought a Metabolism platform to Qatar, a Biomarkers platform we had briefly here.
Yes.
So there was like this combination between logistical and testing, then bring it into studies. And now what Ania is doing, maybe you can continue on that one, is now how can we integrate this really? Because the question in the aim was not to integrate multi-omics at the start. That's where we are now.
Yes, exactly. Because for me, it's like, because for me, I'm biologist, right? So I'm looking from different perspective of the data, and I don't like to look at the Excel sheets, right? You are having p-values, okay, you understand them, but what they really mean. So I was always coming and kind of bugging Karsten with questions. Can we do this? Can we connect? And then he was coming with the idea of this the mutual best hits, right? To connect the data in the first thing, and then this is the way how it started, right? So from the mutual best hit, we were starting to create the interaction matrix between, first of all, the molecules, but also different omics platforms, and this is how we thought. Hold on, I mean, usually you are having the single layer, but here we are having like 18 different platforms. So you are kind of describing the human body with this case control study on the very much molecular level, where you are super specific. And this is the way how we thought about molecular human. So it's coming a little bit from Leonardo da Vinci, the print where you are having the Vitruvian Man, right? Where you look at the ideal way of presenting human being. So we were thinking that with all of those platforms, we are able to create the molecular representation of the human being, right?
Yeah, and what's maybe also important is, I mean, I'm a bioinformatician, so there's also like kind of algorithmic stuff in there. Together with my colleagues, actually also from Munich at the time, Jan Krumsiek, he's now in Cornell in New York. So he was the guy who basically brought Gaussian graphical model, or what is called partial correlations, to metabolomics. And then later he actually used it also on Glycomics data. So in this molecular human, we have like a way of integrating data inside of the platform using these Gaussian graphical models. And then what Ania mentioned, the mutual best hits across platform. So the idea with the mutual best hits, briefly, is an idea that comes from basically orthology in bacteria, where you say, if I want to compare the function of two genes between two bacterial species, I want that the best blast hit in that case from one genome to the other is the reverse best hit from the other genome to the first. And with the same idea, if I now do a mutual best hit between like a glycan, or let's go for, like a glycan and a metabolite, I would only link those that are really like mutual best hits and say these have something in common. So if they are from a similar platform, they could actually be the same molecule if I compare SomaLogic to O-link or Metabolism to Biocritis. But they could also be something, they are basically carrying the same kind of information, although they may be something different. And in this sense, the molecular human and then the web server that we call the Comics server behind that, and maybe we should talk about that in a moment, is basically a tool to actually look at this integrated data, because I don't think you can integrate data automatically. You need to bring it together, put it into a visualization tool, and then you have to use that to interpret it.
Because interpretation is the biggest problem, right? If you're having so many data, what you are going to do with that, right? So what will be your next step? What you see? How they are interacting? This is all the questions which we were asking while working on this project, because I mean it's not something you are doing over the night. So we were having 1,000 discussions and back and forth, and then understanding of how those molecules are interacting, but what these interactions really mean. So our initial goal was to see if we can replicate whatever was previously shown in the literature, and then going further, what is new. But what is key to this entire project is connecting omics, which is our comics server, right? We are seeing it like a window to the molecular human. So there you can have a look on the processes, the interaction between the gene and the molecule, and methylation, for example. It's also linked with the GWAS catalog. So it's pretty robust tool, which is available for everyone. So whoever is interested with some protein, and it's usually looking in the silos, not considering the rest of the processes which are ongoing, can right now go and check. And I would strongly encourage people to do so, because for me, being biologist, right, it's great fun, right? You're having your protein or metabolite of interest, and you can understand how it's connected in the entire network. And I see it as a great plus for the entire scientific community. That's why, I mean, we are sharing it and not keeping it for us only, because we would like to, give the people opportunity to experience what we were experiencing while working on this project,
Just to clarify, you said you were using 18 platforms, and how many omics were you looking at?
I think it's 8. So if we're thinking genomics, then it's methylation, so epigenetics. Then we have transcriptomics, microRNA. Then we had proteomics, glycoproteomics, glycomics, metabolomics, and lipidomics. Yeah, so 9.
So some of the platforms were used for the same omics?
Yes.
Correct.
I mean, the thing is, like, you cannot really draw a line. For instance, would you call Biocritis data, they have a lot of lipids on there, but also metabolites, or Nightingale. Do you call them the one or the other? So I think there are 18 different platforms. And then also we have data in urine for some of them, and in saliva as well. So in the end, there are 18 really different measurements, and we have 2 more that are presently in the making. So we have also mass spec proteomics from the Seer company coming, and from Syngenix auto antibodies against bacteria. But that's still not published yet.
And it's not in the server yet.
Yeah.
But for instance, if you ask about the number of platforms which were used per omics, for Glycomics, we have 3 platforms, right? So, of course, we were checking how good they are correlating and stuff, and the results which we achieved were very, very good. So we're happy with the correlations which we saw.
I'm curious, what was the response of the scientific community or collaborators when you released Comics?
Well, it's pretty fresh. I mean, officially that it really has the name Comics and the molecular human. That's like maybe a year that Ania is working on the paper, giving it names and things like that. The server, I started it during COVID, actually, when I was just blocked and just wanted to do some coding. Before that, I mean, we already shared the data in many studies, so it was used in different GWASs, and we also have a paper together with Gordon Lauch where we compare or combine SomaLogic with the end glycans, for instance. So this kind of integration. So it's not something direct, and there's many colleagues mostly from Cora, that very often come to us and say, hey, you have data on this, can you replicate? Or also with the Twins UK, we're working on replicating things. So the QM-DIVE study is a little bit too small to be a standalone thing for big GWASs or something like that. But if you want to do replication, the nice thing is also it's not Caucasian. There are Indians, there are Filipinos in there. So whenever you see something in QM-DIVE, you know it's really robust. And that's also something which I find interesting, because people would initially criticize it, like say, hey, look, these guys are not all super fasted and they are three different ethnicities and whatever. But the thing is, if you replicate in that cohort and it replicates there, it means that the signal you're talking about is a signal you can carry forward in other studies. And on that side, it's positive.
I think I can also add to the comics as a server, because for the first time we're presenting the comics as a server on the third human glycom project, right? It was the conference which took place in Split, and people were really very positive about it. I also showed to some of the medical students of ours, and they are all like, wow, really, it's possible? Can we look from that perspective? Because usually it's all about the perspective, right? With this tool, we are kind of going out of regular paper and regular publication to another space, to another dimension, where we are letting people to interact with our results. And I think this is something very unique and cool. And this is the way how people, which I was talking about with, they were also seeing it as kind of very cool way of showing the data and putting as a resource this kind of data, because it's not a big deal to bring a table, right? But here you are having much more than that, and this is why I think it was not only simple coding, you know, because Karsten is saying, well, I wanted to code. I mean, no, it was not like this. I mean, it's something more, and something which is really worth it to go to interact with and see how good it is.
Yeah, and I also think it's there. I still see it also a little bit as a pilot for bigger studies, especially when I now see UK Biobank with 300,000 samples from Nightingale. Of course, we cannot compete with that, but in QM Diet, in the comic survey, you can see how the different platforms are complementary to each other, what correlates, what, and you have it. And I think you can also use it to actually evaluate what would you be able to get if you go to a 10 times bigger study like Cora, or a 1,000 times bigger study as the UK Biobank. And I think it could be very motivating as a driver. And I think I even shared very, very long time ago some of the Nightingale data with them to see how does Nightingale compare in terms of coverage to Biocritis, to Metabolon, things like that. So you know the complementarity, and also when you discuss what do you want to do, it's not like, oh, there's just one Metabolomics. I mean, there are 3, 4, 5 platforms, and they are all very, very different. And in the comics server, you can actually see this complementarity and also the similarities with the mutual best hits. You know what is actually overlapping between the platforms and what does each platform has on its own.
So in the molecular human study, you describe glycans as intermediate phenotypes that provide insight into both the genetic background as well as the potential mechanistic link to clinical outcome. Could you explain what is meant by the intermediate phenotype and how it might be advantageous in a clinical setting?
I think the concept, I still give credit to Florian Kronenberg, who is a colleague for me, who was on our first GWAS paper with my Metabolomics in, I think, 2008. And in that study, we really saw this idea of intermediate phenotype to say, I mean, at that time you did GWAS between disease endpoints. So you say, oh, this gene is associated, or genetic variance in this gene is associated with the risk of diabetes, but you didn't really know why. And the idea of the intermediate phenotype is to say, on the one hand, you have a correlation with the metabolite and the disease, which is strong. And then you have also a strong association between the genetic variants and the metabolite. So in that sense, it's an intermediate phenotype. And also, there may be many pathways that all go towards diabetes, and depending on which metabolite, and I mean, now I say metabolite, but it could be a glycan, could be a protein as intermediate phenotype, which ones are the ones that are driving this or that pathway. So the idea of intermediate phenotype is really, like I mentioned before, the imaging, the fine mapping of these pathways. So if I have an association, and it doesn't have to be necessarily genetics, it could be lifestyle factors, smoking, things like that. So whatever you have in deep molecular phenotyping is what I would see as an intermediate phenotype. And that is really working out very, very nicely. And that's the idea why we do all these multi-omics things.
I think from the perspective of our publication, we can say that we saw this, for instance, focusylation in context of cancer and different glycan structures. Obviously, this is something which we need to still follow up and look for the further validated, right, and to further look for the mechanisms. But this is something which can be observed, right? So like interaction between the protein, the methylation, the Glycomics profile, and the link with, for instance, inflammatory mediators and cancer. So you see that there is interaction, you see that there is a link, but then the next question will be, what does it really mean? I mean, what are the mechanisms and what is the driver of these changes and how it's impacting, let's say, cancer, right? If we are looking from the perspective of cancer. And obviously to put it into clinic, it's a long way, right? Like with everything. So, I mean, it's a hint.
And I mean, just as you said, put into the clinic. I think really for us, the challenge, also sometimes the frustration, is the translation. I mean, how can we really make sure that what we do here has really an impact and is not just some kind of academic paper that nobody reads in the end. But that's really nowadays the big challenge, how to translate that. And I don't have a very concrete answer. I think there's not one answer to that anyway.
I think that the entire processes which are ongoing, let's say, there is a huge gap, the one which I see, because right now I'm trying to be even more involved in more clinical study, working on cancer with the physicians. So there is gap between the researchers and the environment of clinic, right? So if we are thinking about translation, first we need to overcome this gap, right? How you will gonna be doing the samples. I mean, if you like to do clinical research of the multi-omics, you need to have entire infrastructure. You cannot come and do a research core, right? You need to have infrastructure of machines, you need to have skilled personnel, and then the readout, right? Because, I mean, we are showing molecular human, we are having so many omics layers, and, I mean, interpretation of those data was a challenge for us initially, right? But if you are a medical doctor and you are seeing this massive amount of data, I mean, how useful it can be for you, right? So I think before we are even moving towards the clinic, it will be the way is it will be pretty long way, and it needs to be also thoughtful what everything we're gonna be doing as a first, second, third step to make it possible. Because, I mean, right now, I see that it can be possible eventually, but not next year or not in 2 years, right?
Yeah, absolutely. Sounds like an ideal scenario where we can have all of these tests to have a big picture, and the doctor can understand exactly what's happening in your body through this elaborate system. But obviously, it will take a long time to enable this.
You throw in the buzzword AI and everything is solved. We don't have to do anything anymore. But that's ironic, right?
But maybe just to go backwards for a second, just to perhaps clarify what is the advantage of having a multi-omic approach when compared to perhaps looking at single molecules when we're trying to understand complex diseases.
So I think the idea of having multiple omics, I mean, you want to look at all the steps that play a role. And as you mentioned before, glycans are at the moment, we know there could be a buzz coming, but we don't see them because we don't measure them.
Also like pieces of the information, right? When you're mentioning about those pixels, right? So if we think about piece of information, like one molecule will be one pixel, but I mean, it's not bringing you to the entire picture, right? But how many pixels you need to see this picture, right? And okay, we were saying that all omics are connected, and it's fascinating that the body is functioning with all this complexity, but if one system is starting to get broken, you cannot really look on single molecule. I mean, even when you look on diabetes, for instance, if you were gonna look on the glucose, it's not enough, no?
And I think it's a trade-off in the end of the resources you want to spend and what you can see, right? And I think for that, I think our study could be very helpful because it's very deep, but you can get a feeling for how deep do you really need to go for your specific understanding, and then focus on things that may be relevant for the one or the other phenotype you're looking at.
Now, you have built this molecular network. Do you think it can potentially give you hints towards molecular interactions which are important in certain diseases but haven't yet been explored in that context?
To some extent, yes, and this was something which we were highlighting in the manuscript. So again, with the glycans and the interaction with the immune system or interaction to immune system and cancer, and in context of cardiovascular diseases also and immune system. So those kind of things we were seeing, some of them we were replicating. That's why we know that the signal which we see is somehow robust and not only novel but also true. But again, it will be worth it to make some verification of these findings, right?
I think the weakness of our studies, of course, I mean, it's a case control study of diabetes, but we don't have follow-up like these big UK Biobank and things like that. So I think what we really need in the future is phenotype, disease, incident disease, this kind of things. And that's why I'm very much saying if we can use this to motivate studies like UK Biobank to have even more omics data on that. And then from there on to say, okay, which kind of correlations do I see at time point 0? And which of these are actually correlated to an incident heart attack or something like that, maybe a few years on?
You have spoken about the challenges of translation of this work, but overall, what do you think are some of the main challenges currently in the era of multi-omics?
I would say it all depends on the perspective, right? Because when I look at the challenges, it will be probably different than when Karsten is looking at the challenges. So I can tell you, I see that there is a need for more omics data, right? And also willingness from the scientists, but the preparation for such a study is maybe not still there. I mean, the very important is to collect data in the high possible quality, that the bioinformatician, they don't have to struggle. But I see that the first struggling with multi-omics is starting initially on the sample collection and the experimental design, right? So this is something which should be kind of clarified once forever, with the processes in place that everybody is kind of obliged to follow them in order to produce good quality data. So the first challenge, it's on the beginning. Then the entire bioinformatics is happening, when Karsten can comment on this one, and then we are having these huge data sets, and it's very difficult to interpret. I see still interpretation of the data when you have so many different factors you need to look at. It's kind of challenging. With the comics, we were able to kind of overcome some of the problem, but there are many stories which we could talk about, right? When we were starting initially, the manuscript was huge, because wherever we were starting to look at, there was coming very interesting story for us, but not necessarily for the reader, So structuring your thoughts and making the story is another thing when you are facing this amount of the data. And also how to connect them is another thing. But there are also other things which are happening in between, right?
And maybe, I mean, when you say translation, there are different kinds of translation. There's one level where it actually absolutely happens, and that's genome-wide association studies. So if you take UK Biobank, the pharma companies are putting millions and even tens of millions of dollars into getting proteomics data and maybe other data sets in the future, whole genome sequencing, exome sequencing in UK Biobank, and the pharma companies are paying for that. And they are not doing this for any altruistic reasons, but because they find genetic evidence for drug targets. And that's what it's all about. And I think that's a huge translation that you have there. It's not very much publicized, because what in the end a pharma company takes out of this and what decision making they do on their drug development, that's not something that's in the public domain. But I think that's a very realistic translation, and it measured in the kind of money pharma companies are putting in there. So I would say there is translation there. Translation to the clinic, I mean, there's another misconception maybe to say, I don't think that you would ever have multi-omics measurements on a single patient. It's more about you would like to find in studies the biomarkers that are relevant. And once you have them, then you measure the relevant biomarkers in the clinics. Although in some cases, I mean, genomics, you do whole genomes on cancer patients, right? And I can imagine that maybe at some point you would do whole metabolome on cancer, on tumor samples to find out what their metabolic weaknesses are. And I think maybe, Ania, you had this one project, maybe you can comment on that kind of translation.
Yes, so this is something which we are trying to do right now, because, I mean, if you think about tumor as a metabolic disease and you would like to understand how you can tweak tumor metabolism in order to support the treatment and things like this. So you will have to understand what are the metabolic processes which are upregulated or downregulated in this tumor to find the way, to find the hint. So to translate it to the clinic, you need to have a real patient, you need to have real tumor, you need to have a real way of extracting the samples from the realistic, very small amount of the tissue. And this is something which we are doing right now, right? So if we will be successful in this approach, we're going to have, let's say, pipeline for like multi-omic characterization of the tumors, right? And also characterization them metabolically to attack the specific metabolic dysregulations which you can see in cancer. So this will be the way of adding extra layer. And why we are looking on this one specifically, because there are already companies which are in fact attacking tumor metabolism in patients in clinical trials, which they are going for glutaminase metabolism or fatty acid synthase metabolism, which is, you know, also again very relevant. The question, of course, is if there are some feedback mechanism from the tumor, do we know them? How we can look at them? And this is something where we were studying together with Karsten on the metabolic engineering approach. So this was, in fact, our very first study together on cancer, when we were drafting the grant, and which was awarded by Qatar Foundation, and we were trying to look at how we can tweak the metabolism of tumor to find the feedback mechanism and to attack both pathways simultaneously. And we have very good example with glutaminase, which is also, which was known that some of the cancers are addicted to glutamine. So our question at that time was, but what's happening then? Is the cancer cell is dying or not? And we find out that no, the cancer cell is not dying, and in fact is activating other metabolic program, which, because we were using the time metabolomics, we were able to detect and rationally target the two pathways at the same time. So, I mean, this is the way how we are starting and how it can be leading further on to some translation. Right?
Yeah, and I mean, that would be the thing where you would maybe go with multi-omics into the clinic. If you really have, like, especially in cancer, I mean, each cancer has their own set of mutations. I think for complex disorders that have one cause, you don't need 1,000 parameters. But if you go for cancer, if you really want to understand the background, what's happening in that cancer, to rationalize what are the escape mechanisms of the cancer to the first level, to the second level treatment. So I think especially for recurring and recalcitrant cancers, there, for instance, a multi-omics approach is definitely interesting. And I think the other thing, it would be similarly the monogenic disorders, which is also something we have a lot of here to deal with in Qatar, where you would have really very rare diseases where there's actually not much knowledge there. So even if you would find the variant, to actually understand what's physiologically happening in that patient, and that would be very individualized if you wanted to have all the omics data and then tell the clinicians, look, this is basically what's happening there. And then you would need, of course, a physician who has enough experience to know how can I tweak the one or the other pathway with an existing drug in these rare disorders. So I think that these are the two things where really the omics I could see directly translated to the clinic. But still, the challenge is the measurement, because you need measurement that's really quantitative, reproducible, not something with batch effects that you can do in cohorts.
Yeah, something which is easy. You can put in clinical setting. Yeah. You need to find somebody who's going to operate this machine, because in proteomics, in metabolomics, in Glycomics lab, there are experts which are troubleshooting the machines, which are developing new ways of optimizing processes. But in clinic, you have to have something which is stable. If you think about all other processes which are ongoing in clinic and biochemistry, right? The biochemistry measurements. So this is something which is established since years. there is, I mean, no go on some deviation unless there will be some problem, right, happening. But usually it's pretty stable measurement. In case of metabolomics or proteomics, I mean, it's still not very much, let's say, quantitative or not batch dependent or not humidity dependent and all of other factors which have to be taken under consideration. So processes which are easier need to be developed to be translated, right?
Absolutely. And I'm just wondering, when thinking about currently, when we mention different layers of omics, some of them are more available to us as consumers already, such as sequencing a genome. So I'm just curious, could you explain how useful looking at the genome, an individual genome in isolation without other omic layers would be in terms of prevention and disease risk?
I would say it's already extremely successful, but there's definitely more to come, right? I think especially if you identify things and you know what they are, then the genome is sufficient, right? If you identify a p53 mutation, you don't have to question it. But if you find variants where you don't really know whether they're really functional, whether it's really the variant that plays the role, then you need extra phenotype to validate that they are actually the ones that do it. And that's especially in these more complex cases like cancers and monogenic disorders.
Also, I think another stuff to add to this is if you are looking for the treatment, right, then you need to, of course, understand how this gene is changing the processes which might be optimized, right? So if you have only genetic information, it's very difficult to see the processes, right, which are changing. I'm not saying yes. So then you need to have all the other factors to think, okay, I have this problem, it's causing this particular phenotype. What are the intermediate phenotypes which I need to, or can I tweak with the drugs, with changing in the diet, to make it, let's say, more feasible to live with this particular genetic problem. Right?
Yeah. And of course, the other thing is, I mean, your genome is stable, right? Except for mutations in cancer, you just have one measure for your lifetime. While the metabolomics and the proteomics, they are very much changing on different time scales. It could be fasting, non-fasting on a daily scale. It could be over a few weeks when you do exercise and even longer. So I think what makes it of course also challenging is that genomics data is easy to deal with because it's 4 letters and they are basically stable forever. The metabolome, in addition to all the experimental things, that they may be, even the same measurements could give you different results depending on how you tune your machine. But even if you would have the perfect machine, they still depend on so many extra factors. So the analysis is much more, I mean, you need to put them into context. And it's on the one hand, it's of course nice. If you go for GlycanAge, for instance, the parameter you derive at genus from that, I mean, that's just one parameter, but that parameter changes with activity, with all kind of things. And that's true for every single parameter we measure. And getting a handle of that, it's also a cost factor, right? If I have to do, I mean, there are these studies from Snyder, right, where you have like hundreds of measurements on just one person. That's the other extreme, right? And that's also extremely interesting. But getting this kind of detail from everyone is probably also a bit impossible to be done. And I think what needs to be developed, and that's really the challenge, is some kind of tools. For instance, there's this glucose monitor that you can put on your arm. If you would have something that would read out maybe 20 or 50 parameters, and that could be a combination of glycans and metabolites and proteins, and we choose the right one. I mean, that, for instance, would be something that would be like the Star Trek thingy, right? You point it to a person and you get like a comprehensive understanding of what's happening. I think that's certainly the dream where we might go one day. And I think that's the path we are on. Yeah. And I mean, we should just be realistic with ourselves where we stand. We're on a path and it's moving forward. I think we're making a lot of progress, especially these last years. I think it's pretty amazing time to be in the field.
Yes, I mean, that's for sure.
Yeah. And it's also to move on away from metabolomics. I mean, we started with metabolomics, but now having all the other layers and getting always exposed to new challenges. And also understanding what glycan data really is was for me also a discovery, because initially I thought, I would get something like omics, metabolomics data from Gordon, but then I realized, oh, this is just one spot on one protein where I have already like 40, 50 parameters, and it's just one, right? And the other day he was just offering me 2 more proteins, so I hope we have opportunity to maybe generate that data as well. And that's then 3 proteins, but I think there are at least 100 or maybe even 1,000 proteins in the bloodstream that are glycoproteins that would be interesting to look at with this kind of tools.
Absolutely. I think a nice way of kind of, I guess, concluding this conversation would be to maybe talk about your efforts currently in the work of translating your research into the clinical application.
Should we say that's classified?
Well, no, I don't know, but I think that the study which Karsten was doing together with our colleague Shaza on the subtypes of diabetes, I think this is something which is pretty clinically very much relevant. No?
Yeah, I think that's an example. That's also an example of how things fit together, because it's not something we did, right? I mean, there is a very highly cited paper from Alquist et al. That came out in a New England journal, I think like 2-3 years back, or maybe 5, and it's very highly cited, and clinicians are jumping on that, and they define basically 4 subtypes of type 2 diabetes. And what we did is first of all, we tried, does the same thing apply to the Qatar population? And it does. And there are other studies showing it in other populations. So it's really something global to humanity and not just for Caucasians. And then we tried to do metabolomics and proteomics on them. And you can find there's really very specific metabolites and proteins that basically go with these subtypes, and you can understand how these subtypes work. And now we're just trying to figure out, I mean, there are people already doing clinical trials to say if you are a certain subtype, is a certain treatment for you better than another one? And that's, of course, the thing where you realize how much time all this takes, right? And once you come up with an idea, if you really want to bring it to the clinic, someone says, oh, clinical trial. And then for me, it's, years, right? Even if you get the money. I mean, it takes you years to get the funding, then it takes you years to do the study, and then you end up with something that in the end you would say, was evident from the beginning, but before you can really bring it to the clinic, you still have to go through that. And I mean, I'm still amazed how that worked with the COVID vaccination, that we really got that in 1 year. It would be nice if we could accelerate the translation of other things as well, but of course the urgency is not the same as for COVID.
Yeah. From my perspective, I'm very much focused on cancer, but this is something which we were just discussing, with the tumor multi-omics from the tumor, how those things are linked, how this is linked to the phenotype of the patient. I mean, all of those questions I'm asking in the study, which I'm currently working on. So this is something which is work in progress, and I hope it will be kind of soon out.
Yeah, I mean, you always ask yourself as a researcher, why are we doing this, right? And I mean, on the one hand, it's just your natural curiosity, it's basic research, it's nice and geeky. But then when I talk, like, for instance, to my mother and say, okay, why are you doing this? I mean, then you really have to say, okay, it's pretty tiny what we really do. We are not, I always think of research as a pyramid. I mean, there are a lot of people at the basis doing a lot of stuff. They are very good researchers, but then they're never in the limelight, and they're all working together. And in the end, you have the few who are maybe just lucky to develop the RNA vaccine or things like that then really make an impact. But I tend to feel that they wouldn't have done that with the whole community around that, and we are part of that. And that's what I think what science is really about and what makes it attractive to me.
Yeah, that's how it is. Because I mean, on the end of the day, I mean, my motivation is always to think how we can help, right? But I'm not so naive as in the past, because many years ago when I did my master, I thought that with the method which I was gonna be doing, I will be solving the cancer problem, right? Because we were working that time on non-viral gene delivery, and this was something, well, for sure it will gonna work. I mean, not everything is always like, I mean, it was working, right? But you know that you need to have so many different things on the way, and of course with time, you like to contribute. This is the most important, and of course making some kind of progress. And I hope we'll be able to do it with the things which we are doing.
I will certainly continue to follow your work and find out more about these new studies which are somewhat classified still.
Thank you.
All right, thank you.
Thank you very much. Now speaking to our listeners, I hope this conversation gave you an insight into the tremendous potential that multi-omics approach holds for the future of personalized medicine and human health. If you would like to access more information about this conversation, as well as about Ania and Karsten's previous work, follow the link in the description to the show notes for this episode. Equally, if you would like to find out more about GlycanAge, head on to glycanage.com, where you can access a whole list of our scientific publications, blog posts, testimonials, and of course, this is where you can order your GlycanAge test kit. Watch out for our next episode, where I will be joined by Ronald Schnarr, a professor of neuroscience at Johns Hopkins University, whose research focuses on neuroglycobiology. We will explore the role of glycans in the healthy structure and function of the brain, as well as what glycan research can tell us about neurodegenerative diseases, such as Alzheimer's disease. Please don't forget to leave ratings and reviews for this episode and engage with us on social media. Thank you for listening and have a great day.